Welcome back to another Year 13 Biology revision video, and we're going to continue with respiration. We're going to look at our second lesson, which is on the link reaction and the Krebs cycle. So I thought I'd just quickly recap some of what we learned last week in glycolysis. So in glycolysis we converted glucose into two molecules of pyruvate, and we're going to follow the fate of that pyruvate molecule with these two metabolic pathways. We generated a little bit of ATP as well in glycolysis, and we reduced some NAD as well. So all of glycolysis took place inside the cytoplasm, and that can occur if oxygen's present or not. It doesn't really matter. These, however, do take place within the mitochondria. More specifically, the link reaction and the Krebs cycle take place inside the matrix of the mitochondria. Okay, so this only really occurs in eukaryotes that have true mitochondria. So these have the pyruvate that was generated in glycolysis is actively transported into the mitochondrial matrix. So we have to use a little bit of that ATP, a little bit of that potential energy to move that in. Once it's inside, we can then begin the first of these two processes, the link reaction. So I'm going to cut now to a diagram I created on PowerPoint of the link reaction, which hopefully give you a nice overview of what's happening. So what's going on? So we start off with this 3-carbon compound pyruvate, and then what we're going to do is we're going to remove some carbon dioxide from it in a process called decarboxylation, which is done by an enzyme called decarboxylase, not too surprising. We also are going to reduce some NAD as well, so we're going to remove 2 hydrogen atoms away from our pyruvate, and that will leave us with a different molecule, a 2-carbon molecule now, because we've lost that CO2, remember, so we're down just to 2, and that is called acetate. Now acetate will combine with another type of coenzyme. This one's called coenzyme A, or CoA if you like, and that will form a complex with the acetate, which we call acetyl-CoA. Okay, so that's it for the link reaction. It's that simple, effectively. So we don't generate any ATP at this stage. This is the first sign of any carbon dioxide being produced in our respiration, which hopefully you remember from GCSE, that equation. And we reduce a little bit more NAD. So that acetyl-CoA complex goes on to the next stage of aerobic respiration, which is called the Krebs cycle. And again, I'm going to cut to that diagram I created and talk you through that one. So the Krebs cycle, I guess in some ways, might look a little bit superficially like the Calvin cycle, as in it's a cycle, it involves a series of enzyme control reactions. So in that respect it is similar, but this time instead of like trying to build up and make something like glucose, we're actually further breaking down molecules, and it involves a series of oxidation and reduction reactions to basically create more reduced NAD and another coenzyme that's going to crop up, reduced FAD. Okay, so that acetyl-CoA will come into the cycle, and it will combine with what we call an intermediate molecule. So it doesn't really have an actual kind of primary role itself, it's just involved in that cycle. So the acetyl-CoA will combine with that 4-carbon molecule to make a new 6-carbon intermediate molecule. The CoA is not used up. That coenzyme then can go back to link reaction and take up another acetate. Okay, so that can be constantly regenerated and recycled like all coenzymes. They're never used up. So that 4-carbon molecule will be converted back into its 4-carbon state by removing 2 carbon dioxide molecules, so some more decarboxylation occurring. We'll also reduce 3 molecules of NAD, so we're going to remove some hydrogen from that substance, and we'll also reduce 1 molecule of that coenzyme called FAD. Okay. In addition to that, we should also be able to synthesize 1 molecule of ATP from ADP and phosphate. Okay, and that we call substrate level phosphorylation. So far, with respiration included, we've seen 2 types of phosphorylation. We've seen photophosphorylation, which is what we saw in photosynthesis using light energy. And here we're seeing another method where we're actually using a chemical substrate to synthesize ATP. There is a third type, which is called oxidative phosphorylation, which is our next lesson in this series on respiration, and I'll talk more in detail about that. So let's summarize this. Let's get a bit of an idea in our head. So acetyl-CoA releases the acetate, which combines with a 4-carbon molecule to make a 6-carbon one. It then loses 2 molecules of carbon dioxide. We reduce 3 molecules of NAD and 1 molecule of FAD, and we synthesize 1 molecule of ATP from ADP and phosphate. So now in effect we've really broken down all of the glucose. It's now been split down into carbon dioxide and hydrogen. Okay, because those hydrogen is now being carried by those coenzymes FAD and NAD, and that's their role. So we've only generated a really small amount of ATP so far. Okay, not very much at all. So most of the ATP will be synthesized in that final stage that we call oxidative phosphorylation, and it's those coenzymes that are going to be really important there. And actually, that hydrogen, those hydrogen atoms, is what contains all that chemical potential energy that we can transfer into those high energy bonds with an ATP. Another key thing to remember as well about the link reaction in the Krebs cycle is that we get 2 of them per glucose molecule. What do I mean by that? Well, if you remember, if we go back to glycolysis and we saw that we made 2 pyruvate from 1 glucose, that means that the link reaction will happen twice for each glucose, because 1 pyruvate comes in, becomes acetate, that happens once, and of course in that second pyruvate does the same thing. So we get 2 runs of the link reaction and 2 turns of the Krebs cycle as well. So you end up doubling everything effectively if you're looking at glucose, or if you're looking per pyruvate or acetate, it's just that 1 round. So for example, when we looked at the Krebs cycle, I said that we reduced 3 NAD. Well, we actually reduce 6 if we're looking at per glucose. Okay, so I've just doubled all those quantities. All right, so that's it really. It's quite simple in many respects, the reaction in the Krebs cycle. The Krebs does look daunting, I know that. Very rarely though do you need to be able to describe it in absolute detail. Having a more general understanding of it and the idea of what the products are, like the NAD, the FAD, the CO2 and the ATP, is a really, really important bit. You might also see in the exams the proper names for those intermediate compounds like citrate and stuff like that. Sometimes we call the Krebs cycle the citric acid cycle as a result. But they will give you the information you need in the exam question to help you. So don't be put off if you see those sorts of things going on. All right, so this is Dr. P signing off. Stay safe, have a good time. Cheers.